uh, TCC-GUI, which is a graphical user interface for a package for RNA-seq analysis. It is based on RShiny web application package. It starts from the data simulation. You can either upload or generate simulation data here. It has a select random seed and number of genes, and then proportion of the differentially expressed genes and number of groups that you want. We select the default one and we generate some simulation data. Okay, that is done. And now we want to go to the next step, which is exploring the analysis. We just import the data, which is done, and then we assign group. The groups, now we have replicate 1 and replicate 2 and replicate 3 for gene for group 1 and the same number of replicates for group 2. We keep the names as it is and we move forward. So we now have a graphs of the original raw count data for the replicates and for different groups. We can have filtering of the based on the numbers of the genes, how many count if you want to filter them out, and a density plot about the distribution of those counts, and Mds Plots, which is multidimensional distance plot. It has different options and you can see how different replicates are characterized in different regions of when you do this MDS analysis. Then you go to the PCA plot and you see the different principal components, and you can have as PC principal 1 and 2 in 2D plot, and with these 3 3D components in the third one. After that, we can go to the actual differential analysis, differential expression analysis. We can use different methods, which TCC-a is a pretty popular one, to get the differential expressed genes, and we can have filtering and stuff like that, but we go with default, and then it takes some time to do differential expressed gene analysis with TCC-a, and let's see when it's going to finish. And it is done. So we have now the data. We have lots of options for plotting the data, and we have genes and everything. So we can generate MA plot based on this is just for graphical interface and the false discovery rate cutoff. We go with default when we generate the MA plot, which the red one shows the differential expressed genes, and you can see on the right hand side the raw count number of genes. After that, we can go to the Volcano Plot, which you can just see the range that you want to x-axis be fold change and the p-value for cutoff, and we generate the Volcano Plot. So you can see the ones that have highest fold change and the one that has the lowest value of the p-value, so they are most significant. And also on the right hand side you can see the raw counts for different ones. And then you can go to the heat map and plot a heat map based on the, for example, false discovery rate, and then you can run the heat map analysis. So it takes some time to generate that heat map, but you can see the raw data is available, and then the different groups and different replicates and the genes and how they are clustered together. Then we go to the next step, which is expression level. We need to set some genes that we want to see more details of the expression level. Let's go, because the genes are just called gene 1, gene 2, gene 3, and gene 4. We just go with them, and then we generate a plot. You can see for each sample, we can have the number of the counts and the expression level. And so then we go to the report section and we can generate the report. We might get disconnected here, which is probably a bug in the code, but in general it looks very nice and it works perfectly in other sections.